Hello, everyone, and welcome to today's AAPS eChalk Talk. Today's eChalk Talk is hosted by Sapient. Today's eChalk Talk is titled Advancing Clinical Biomarkers Through Mass Spectrometry-based Discovery Proteomics. Proteins represent the primary effectors of human biology, and evaluating the thousands of proteins in the human proteome provides critical insight into cellular physiology and disease mechanisms that cannot be fully inferred from genomic data alone. This webinar will discuss next-generation bioanalytical systems for proteomic studies in humans, focusing on high-throughput discovery mass spectrometry proteomics as an analytical solution that now enables accurate and robust measure of thousands of proteins across thousands of individuals, while providing highly accurate protein identification and measure of post-translational modifications. To kick things off, I'd like to introduce our speaker, Dr. Mo Jain, founder and CEO of Sapient. Dr. Jain is a physician scientist with more than 20 years of expertise in physiology, computational biology, and mass spectrometry-based bioanalytics. He formed the Jain Laboratory at UCSD, where he led his team to develop next-generation mass spectrometry systems to probe the non-genetic landscape of disease across large-scale human studies. Dr. Jain founded Sapient in 2021 as a spin-out of the Jain Laboratory to expand upon his mission, providing bespoke services for multi-omics biomarker discovery. I think you'll find his presentation topic very timely. So without further ado, Mo, please take it away.
Thank you so much, Stacy, and really welcome to all the listeners today. We're really excited to spend the next half an hour or so talking through dynamic biomarkers, particularly within the proteome, and emerging technologies that now allow us for the first time to integrate and interrogate this area of biology and some of the discoveries that may emerge from it. So to begin with, the way we think about human disease is really as a dynamic process. There's a continuum or spectrum from health to illness, and we can fall anywhere along that spectrum. When we think about that transition from a healthy state to an illness or disease state, you can imagine there's several critical points along that trajectory, whereby we may develop acute illness. Oftentimes in the setting of an acute illness, we recover. And for chronic illnesses, there's this long indolent decline whereby disease is instituted at its earliest time points, years or even decades prior to actual clinical manifestation. Being able to diagnose disease at this earliest time point and understand the key druggable targets is essential for curing disease. As we go down this trajectory and we decline from a healthy to a disease state, disease becomes clinically apparent and manifests at some point. Oftentimes, patients present to a doctor's office at that time where they're diagnosed. And we know clinically that patients, even that carry the same diagnosis, can have very different clinical outcomes. There's some individuals that have a slow, indolent response to disease states where they may decline very slowly over decades, whereas other individuals have a much more precipitous decline in their clinical state over time. Being able to identify that prognostic state at the earliest time point is essential for stratifying individuals and best aligning them with a particular therapeutic. Oftentimes, we know drugs are instituted at this point, or pharmaceuticals, in order to improve the clinical state. And we once again know in certain individuals, they benefit from these agents, whereas other individuals continue to decline despite these therapeutic interventions. And in the same way, we need to be able to diagnose disease at its earliest state, understand prognosis. We also need to be able to guide in a precise manner the way therapeutics are instituted, whereby we align patients with their specific disease state and the best drug for that disease process. Now, given this dynamic nature of disease, this requires dynamic measures or biomarkers that allow us to read out each of these steps, whether it be the acute illness step or in the setting of chronic disease, being able to diagnose disease at its earliest time point, risk stratify patients, and then ultimately to guide therapeutic interventions. Now, the way we can think about this in the simplest just terms, if we look at a map here of Southern California, what genomics has done for us is to provide the roadmap, the underlying framework for what may to be, for the inheritability of disease and those processes that may contribute to disease. But for anyone who's driven in Southern California knows, it's not the map that's critical, but rather the dynamic traffic patterns that really provide the maximal amount of information, especially if you're trying to go from point A to point B or to understand how you can bypass bottlenecks in the roadmap. The way we think of this is genomics is that underlying map, and dynamic biomarkers, including proteomics, really read out the underlying traffic patterns. Now, there's many different flavors to dynamic biomarkers, and here at Sapient, we focus on a subset of these that we feel are most critical. We view this as our multi-omics biomarker discovery platform, and this includes various modalities, including metabolomics and lipidomics, discovery proteomics that we're going to spend time on talking about today, precision-guided proteomics, where we're measuring a small subsets of proteins in a very regulated manner, and even other dynamic processes such as single-cell and bulk RNA sequencing. Now, in all cases, what's critical is all of these measurements, from metabolomics and lipidomics to proteomics to precision-based measures to bulk RNA sequencing or even single-cell sequencing, can be performed in a singular sample. So all of these have the ability to measure thousands of markers, again, in that one sample, and can be scaled here at Sapient to tens of thousands of biosamples at a time. And this is really what enables that discovery process. When we think about specifically protein and proteomic biomarkers, there's some very unique features of these molecules. For one, we know these molecules are released from tissues into blood, and this allows us to non-invasively make these measurements in a blood sample and reveal what's actually happening at the level of the tissue. These are highly dynamic markers that are reflective of dynamic processes, whether it be this transition from health to disease or response even to a particular intervention, such as a therapeutic intervention. These are highly retained and conserved from cellular systems to preclinical systems to humans. And that allows us to translate in a forward as well as reverse direction between preclinical models or cellular models and human studies. And given the way in which we measure these can be rapidly translated. They're amenable to precision-guided measures under CLIA or CAP-regulated laboratories, which allows us to rapidly translate any discoveries back to clinical trials and clinical practice. Now, the challenge with proteomics as a balancing act is there often is with any bioanalytical solution, in which we're typically balancing the number of molecules that we can assay and the throughput and speed with which we can do that. Now, with proteomics in particular, there's several other nuanced factors that are critical to think through from a balancing perspective. On one hand, you have analytical specificity. When you're identifying a specific protein, is this the correct identity? As well as what we call post-translational modifications, which are secondary modifications that occur on proteins, such as phosphorylation and methylation, that can be critical for the protein activity. We also have throughput, the number of samples that can be processed, and that has direct implications not only for cost, but also the level at which discovery can be made. We know that there's tens of thousands of proteins, either in tissue samples or in blood samples, and being able to capture the maximal depth is critical. And then fundamentally, the biological insight, which reveals the number and types of proteins that we're actually capturing, is quite critical. And so there's this balancing act and a hurdle that we must overcome in order to make proteomics a staple of our biomarker discovery strategy. Now, it's absolutely been proven over the last several years as the first large-scale proteomic studies have emerged from a number of vendors and a number of investigators from around the world, just how much biological insight lies within the underlying proteome. Here are three such publications that were recently presented in the last several years, looking at simple matrices such as plasma, in which thousands of proteins have been assayed, and how much has been learned from this, including early disease detection markers, new mechanisms and new potential targets for disease intervention, et cetera, et cetera. But there's much to be learned as we're able to make these measurements across large numbers of biological samples. Much of the work that's been done today has leveraged what we call affinity-based proteomics. These are methodologies that have been implemented by SomaLogic or by Olink and really serve as the first foray into large-scale proteomics, particularly within the plasma system. Now, what these affinity-based proteomics do is to leverage binding agents. In the case of Olink, either polyclonal or monoclonal antibodies, and in the instance of somologic, aptamer, or synthetic aptamer-based binding agents. And in both cases, what these binding agents are doing are binding particular proteins within the plasma, and then they're being read out by a genetic tag, which is affixed to the underlying binding agent. Within Olink, there's two of these sort of binding agents, two antibodies per target, and within Somologic, there's a single aptamer. Now, these tools have been instrumental and really have opened up the eyes of the world to the amount of insight that can be achieved by large-scale proteomics. The challenge with them, however, really has to do with the underlying specificity, in that the ID of the actual protein that's being assayed is heavily dependent upon the specificity of the binding that occurs between the antibodies or the aptamers and that target protein. Now, anyone who's ever run a gel with a polyclonal antibody or done a pull-down assay with an aptamer knows that the specificity of these molecules, while quite good, is not perfect. And oftentimes, what we think we're being pulled down or what we think is binding is not actually the molecule that's being bound. If we look at large-scale data that's been released, this is from the Decode group in a recent Nature publication, across thousands of individuals in which the same samples were run on the Olink 3K panel and the Somologic 4K panel, we find there's over 1,800 proteins that are assayed on both systems. If we look at those 1,800 proteins that are matched on both systems and measured in the same biological samples, and simply ask how correlated are the measurements, what we find is the distribution plot on the bottom left. Now, you can see that there's a group of proteins to the right here that have a fairly high Spearman correlation, above 0.6, and are highly correlated measurements, which means that the identity of the measurement is correct, the identity of the molecule is correct that's being bound, as well as the actual quantitative measurement is correct. Concerning, however, is what we see here to the left of this graph, and where there's a big peak around a correlation of 0 that suggests that for a number of the molecules that are captured on these two systems, they're not completely correlated. In fact, they could be randomly correlated. The reason for this could be twofold. One, the underlying identity of the protein that's being assayed may be incorrect on one system or the other. Or the other issue that may be occurring is, given the fact that these use affinity-based binding, you could imagine that common variants, structural variants that occur in the protein, or post-translational modifications that occur on the protein may interfere with the actual affinity-based binding. In that case, you may have an alternative or a false measurement that has nothing to do with the protein levels, but rather do with structural changes in the protein that occur from, again, genetic variants or PTMs or other binding events. So if we look at these two platforms on the bottom right, we find that there's somewhere in the order of about 500 proteins on each of these that are what we consider validated in the literature, meaning that there's a pQTL, meaning a genetic interaction for which we can be reasonably certain about the underlying identity of the protein, as well as these are highly correlated between these two platforms. There's another 2,000 or so proteins for which there's what we call cis-pQTLs. If we look at a genetic variant in a protein and we look at that protein measure in the blood, those are highly correlated, which suggests that the identity of the protein, and once again, the measurement, is quite well done. And for both of these platforms, you can see that there's a dark blue area here for which we do not have pQTLs, and we do not have evidence for external validation, which again calls into question whether or not the identity of each of these is correct and whether or not the quantitative measure is also correct. I want to be clear, though, that these methodologies really were the first to be able to be deployed given the underlying throughput across large population scale studies. So despite some of these challenges, these have really been at the forefront of the discovery process for the last several years. Now, what the secondary question that emerges from this is there a way to bring discovery proteomics into balance, to align the analytical specificity in PTMs, as well as the throughput and cost, the depth, meaning the number of proteins that can be assayed, whether it be in a blood sample, meaning plasma or serum, in a tissue sample, in a preclinical sample, as well as a clinical sample, and in doing so, maximize the underlying biological insight. And this is where Sapient has spent much time developing what we consider our high-throughput discovery mass spectrometry platforms. Shown here is a video of our laboratory, again, based in San Diego, California. And what our systems do and what our pipelines leverage is state-of-the-art Bruker-Timstorf proteome mass spectrometry. And what these systems allow us to do is to measure thousands of multi-omic measurements, whether they be metabolites, lipids, and for the sake of this conversation, proteins across multiple sample matrices, from liquid samples such as blood and urine and CSF, tears and eye fluid, to solid state samples such as tumors and tissues, cellular material, and even media, across both preclinical and clinical samples. Each of these measurements is amenable to precision-targeted panels in which we can take a discovery and bring it into our CLIA laboratory in order to develop a regulated assay that may be deployed then as part of a clinical trial or for clinical translation with rigorous QC. Now, this provides an overview of the pipeline for proteomic discovery and how we approach this, particularly within the context of plasma. So plasma, as anyone's aware, has a very high dynamic range for the circulating proteins that are present within that biomatrix, which means that the top proteins, the top 22 or so proteins, account for 98% of the total biomass. And what ends up happening with mass spectrometry is that those top abundant proteins swamp the underlying signal and makes it very difficult to measure the less abundant proteins. Now, in order to be able to overcome this challenge, one needs to deplete the top abundant proteins while enriching for the less abundant proteins. And this has been a real challenge over the last several decades. The way we approach this here at Sapient is through a use of a nanoparticle capture system. These are hollow particles that we can introduce into a plasma sample. And what these allow us to do is to bind, given the charge state of these particles, free-floating plasma proteins, as well as capture exosomal, extracellular vesicle, and membrane-bound proteins that are within our circulation within the inside of these nanoparticle systems. If we look at raw plasma in which it has not been depleted, we can only measure 200 to 300 proteins using mass spectrometry. If we look at depleted plasma, that means plasma in which we've removed the most abundant proteins only, we can measure somewhere in the order of 800 to 1,000 proteins. Now, if we look at nanoparticle enrichment, again, in which we're depleting the top most abundant molecules and enriching for the lower abundant molecules, this is where we're now able to measure north of 4,000 proteins in PTMs in a human plasma sample or in a preclinical plasma sample. For all of these systems, we're enriching by nanoparticles and then leveraging Bruker-Timstorf mass spectrometry for label-free DIA discovery proteomics. And the way these systems work is a protein is broken down into its core peptides, and using trypsin and lysine and other digestion materials. And then we're directly sequencing those underlying peptides, and from that then reconstructing the protein abundant levels. When we do this, we have multiple measurements per protein, which means we have very high confidence in the underlying protein identity. This is the underlying specificity of the measurement, as well as this allows us not only to measure the protein itself, but the post-translational modifications that are on that protein. And this is one of the limitations of affinity-based approaches in which the antibody can just measure the native protein and less so the actual PTMs. For the proteomic space, these PTMs are absolutely essential and tell us a lot about the underlying functionality of a protein. When we look at human or preclinical blood, there's a number of these PTMs that we're interrogating, including phosphorylation, acetylation, methylation, and ubiquitination. Within tissues and cells, we can also capture sumylation and other critical PTMs. As I mentioned earlier, all of our approaches are amenable to multiple liquid and tissue biomatrices, not only plasma and serum, but also urine, CSF, tears, eye fluid, and media, as well as tumor and tissue and cellular material from both clinical samples, meaning human samples, as well as preclinical models. And critically, because we're capturing not only the free-floating plasma proteins, but also the exosomal and EV-bound proteins, we're measuring most of the biologically important proteins, including over 1,000 exosomal proteins and over 1,700 membrane-bound proteins from various tissues within the blood circulation. Now, even within this proteomics approaches, there's no singular solution, and there's many, many flavors in which we're trading off the underlying throughput relative to the depth of measurement, meaning how fast we can go and the number of samples that can be analyzed versus the number of proteins that can be measured in any one biological specimen. And for this reason, we have 4 different offerings within our in our plasma proteomics approaches. To the left, we have what we call our high throughput discovery approaches. These are modalities and approaches that are really optimized for throughput and robustness across large human studies or large preclinical studies. Underlying these systems, we can capture somewhere in the order of 1,800 or so proteins in PTM, typically in plasma. And these projects, we have enormous capacity that enable us to study more than 100,000 samples in a given biological experiment. We have next what's called our core discovery method. All of these use the same nanoparticle extraction as well as the same mass spectrometry approaches. And this core methodology is really optimized for revealing biological insights within the plasma proteome. This methodology captures more than 4,000 proteins in PTM and plasma, whether it be preclinical or human plasma. And this is amenable to projects from 10 to 10,000 or so biological samples. For cells and tissue, we have what's called our deep discovery method. And these are really optimized for when the deepest coverage of the proteome, measuring the maximum number of proteins, is really what's desired. And this allows us to measure more than 10,000 proteins in PTMs in a singular sample and is amenable to projects up to 1,000 or so biological specimens. And all the way to the right here, we have what's called our precision proteomics approaches. After we've made a discovery and we know what are the key proteins, we can develop bespoke assays for targeted quantitative measure of these key proteins of interest, whether these are from a prior hypothesis or emerge from the discovery that we perform on behalf of our clients. These can be quite high throughput, from 1 to 10,000 or so biosamples as part of clinical trials or clinical implementation. And most importantly, these robust measures can be done under CLIA designation, which allow us to implement them as part of clinical practice. Now, everything we do here at Sapient is really guided by this concept of translation. How do we enable discovery for our clients? We operate as a service CRO, and so really not only being able to make the initial discoveries, but translate these discoveries on behalf of our clients is absolutely essential. All the way to the left here, we start with discovery, where we're making multi-omics data measurements, whether it be around metabolomics and lipidomics, RNA sequencing, and proteomics, which we discussed here today. We're providing not only data generation, but biocomputational analysis of the underlying data, where we can do multidimensional data integration and use advanced statistics to identify what are the key biomarkers that answer a specific question. How do we drive actionable knowledge and insight? We believe very strongly in validation, both technical validation using orthogonal methodologies, as well as biological validation using Sapient's human biological database that I'll talk about in just a moment. And then ultimately, as I mentioned, for any of the measurements we do here at Sapient, we can rapidly develop a CLIA assay that allows us to translate these singular discoveries back to clinical trials and clinical practice. Now, as you can imagine, these tools and technologies can generate massive, massive amounts of data. And if there's any one lesson we've learned over the last several years is that data in isolation is of actually little value. And the goal is actionable knowledge or insight. Information that answers a biological question that allows our clients to drive forward a drug development program. So how do we bridge this gap between data and knowledge? And this really comes from bioinformatics analysis. So as I mentioned earlier, here at Sapient, we provide not only bioanalytical services for generation of proteomics data, but also extensive statistical analysis of that underlying data around specific questions that are of greatest interest to our clients. Everything from biological and pathway signaling, what's called pQTL and MQTL analyses, genetic interactions, identification of new targets and target engagement, mechanisms of actions for various therapeutics, as well as clinical measures, including early disease metrics, disease progression markers, and companion diagnostics. And in doing so, this is how we can help our clients bridge this gap between data generation and discovery and actionable knowledge that moves forward a drug development program. Now, here's one such example. If we look at the plasma proteome in a typical human biological specimen, a plasma sample, there's over 40 different signaling pathways, tissue signaling pathways, that are actually captured within the plasma biosample using our proteomics approaches for the core method. This span over 9 key biological processes, everything from mixed signaling and PI3 kinase signaling to inflammatory signaling cascades and interferon signaling. And in doing so, from the blood sample itself, we can derive actual signaling cascade activity measures that may be occurring in the local tissues. You can see for each of these signaling pathways, there's anywhere on the order of half a dozen to several dozen different proteins that are represented in each of these pathways that are present within the plasma proteome that allow us to interrogate the actual activity of these specific pathways. In the same way, there's proteins from over 28 different tissues that we're interrogating within the plasma, both in the free-floating plasma proteins as well as in these exosomal and membrane-bound proteins that can tell us about tissue physiology and injury and disease processes that may be occurring specifically within those tissue beds. Now, as I mentioned earlier, part of this translation process requires other underlying data assets that allow us to take a discovery and amplify the value of that discovery. And in doing so, we've built what's called Sapient's Human Biology Database. This is a database of over 100,000 human biological specimens that have been collected from around the world, in which we have extensive human clinical phenotypic measures across over 100 different disease states that are shown here on this pinwheel to the right. There's over 130 different million phenotypic data points relating to genomic measures, longitudinal clinical outcomes over 10 to 30 years, lifestyle factors and measurements, response to various drugs and therapeutics, demographics, microbiome, and even protein measurements. And on each of these biological samples now, we're layering on multidimensional multi-omics data, both metabolomics and lipidomics, as well as proteomics using our underlying approaches. What this allows us to do is to take any discovery for our client and quickly extend that discovery to other disease states and other time points in understanding how those biomarkers may be playing a role in human biology and disease. Now, I just want to take the last several minutes here before we open up for questions and talk about potential applications. And there's many, as you can imagine. These are fundamental tools that can be applied in different ways. One of the ways we think a lot about is how discovery proteomics may be used in the development of antibody-directed drug conjugates, as well as for T-cell engagers. These are agents that are signaling or binding particular tumor-specific antigens or tumor-associated antigens on a tumor sample and either introducing a various chemotherapeutic within that tumor cell or calling in immune cells to target that specific tumor sample. Now, in order to sort of aid in the development of these ADCs or T-cell engagers, discovery mass spectrometry can do several things. It allows us to measure membrane-bound enriched proteins on a tumor sample, and in doing so, identify tumor-associated antigens, both in cell lines and tissues that are critical for developing of the binding characteristics of these ADCs. We can measure over 1,000 surface proteins, either in a tumor sample or in cellular samples, and once again, in doing so, identify relatively unique tumor-associated antigens, both in tumor versus normal, as well as across different tumor types. Given the fact that we're doing label-free DIA proteomics, this allows us to very accurately quantify the protein copy number across different TAAs, and in doing so, once again, identify what may be the best target for an ADC-based therapeutic. At the same time, we work quite a bit in the PROTAC and the molecular glue space, in which we're using proteomics to do a couple of different things. When we're looking at these degrader systems, we can be able to measure thousands of proteins to comprehensively assess the specificity of the degrader. Is the PROTAC or the molecular glue degrading selectively the protein of interest without artificially degrading other proteins? We can do very high throughput measurements across hundreds to thousands of compounds around selective targets of interest, and in doing so, screen large compounds for new degrader agents. We have methodologies that allow us to do protein turnover and resynthesis kinetics, or what's termed the protein half-life, again, to understand how these molecular glues and degrader systems may be most efficacious as a therapeutic intervention. And then fundamentally, we can monitor changes in the cellular proteome over time that may explain how these PROTAC agents or molecular glues are functioning or contributing to drug resistance or adverse events over time in individuals. Now, when we think about the differences between how Sapient approaches proteomics and others, there's several key factors that we reviewed here today. First, our systems allow us to measure a large number of proteins, more than 4,000 proteins in plasma, and more than 10,000 in cells and tissue. And this is really performed in a discovery manner. So not specifying the proteins, but rather asking the mass spectrometers to measure everything that may be present above a certain concentration in that biosample. We're able to measure both PTM and pressure and protein variants through peptide sequencing, and this is essential for understanding how the proteins may actually be functioning. We have very high confidence in protein identification because there's multiple peptides per protein that are being sequenced, and we perform not only the bioanalytics, but also bioinformatics around this data in a manner that allows us to advance forward actionable knowledge. These tools can be applied across multiple liquid and tissue samples, both blood and tumor, normal tissue and cells, media, etc. And these are highly scalable across multiple samples, up to tens and now hundreds of thousands of biosamples at a time. And we can do this in a very cost-effective manner with very quick turnaround time. There's many different protein markers that you can imagine, everything from targeted identification to companion diagnostics. And we work across many different therapeutic areas as shown here on the right, again, that allow us to enable discovery for our clients. I'll go ahead and stop there, time allotting. I'd be happy to take any questions, and thank you for the audience for their attention and time today.
Thank you so much, Mo. That was really a great presentation. And we do have a couple of questions that have come in, and I do encourage our audience to keep submitting questions. We will stay to answer them. So the first question, can you elaborate on the PTMs and protein variants that your method measures? What role do these protein modifications play in protein function and disease, and how can PTMs be targeted for therapeutic intervention?
Absolutely. It's a great question, Stacey. Thank you for it. And thank you to the audience for raising this question. So the way we think about proteins and the way proteins function, there's two critical measures. One is the abundance of that protein. Is the protein there and how much of it is there? And the second is what you refer to again as these post-translational modifications or PTM, Stacey's, which is in the way the body or the cells turn on and off and alter the underlying function of an actual protein. So you can imagine to interrogate these biosamples in a way in which it provides maximal information, we need to be able to measure not only the levels of the proteins, but also these PTMs. Now, there's many different types of PTMs that occur in both preclinical and human samples that are regulated by the cell for turning on and off these proteins or changing their function. This includes phosphorylation, acetylation, ubiquitination, methylation, somylation, et cetera, et cetera. And these PTMs can all be captured using our discovery approaches, either through our deep discovery broad screening or through enrichment using selective nanoparticles and antibodies that allow us to specifically pull out molecules that have a specific post-translational modification. Now, you can imagine from a drug perspective, answering the second part of your question, Stacey, the way we target this is through targeting the proteins that add or remove these PTM from proteins. And so, for instance, kinases, which are a very active area of target validation in the oncology space, have been essential as targets. And these add on phosphorylation or remove phosphorylation signals from various proteins. And so we can target the mechanisms that add on or remove these PTMs from various proteins.
Great, thank you. Another is, you mentioned that your method specifically captures membrane and exosomal proteins. Can you explain how this works and how it's different from affinity-based proteomics approaches such as Olink or SomaLogic?
Absolutely. So you can imagine for affinity-based approaches, these are really optimized, again, because they're using binding agents for capturing free-floating proteins within the plasma sample. And they do it really quite well for the most part. And that's why we view our mass spectrometry approaches as complementary to these underlying affinity-based issues or affinity-based methodologies. Now, when we think about membrane and exosomal proteins or what we call extracellular vesicles or EV proteins, these are proteins that are shed from cells that are typically still living, that are budding off, and then are traveling through the blood. And this is one of the main ways in which various cells communicate with one another. You can think of it as the language channel for these cells. So being able to interrogate that language, both to understand when a cell is in trouble and it's diseased or it's damaged or it's signaling to another cell in a body, is really fundamental. Now, the way our nanoparticle systems work is we're capturing not only the free-floating plasma proteins, those that are in circulation, but also these exosomal fractions and these extracellular vesicles or shed membrane proteins that are within the blood. Now, oftentimes what we find is that these can be absolutely essential for revealing early disease processes in various tissues and allows us to non-invasively understand what's happening in a tissue from the blood sample. In other cases, such as in oncology with an ADC drug, measuring these membrane-bound proteins in blood can provide quite a bit of information regarding target engagement of a particular therapeutic, or can even reveal new potential targets for engagement as part of these directed therapeutics. And so being able to capture these molecules is really, really critical. We do this through nanoparticle-based enrichment, and this is slightly different than the affinity-based approaches that are really geared towards the free-floating plasma proteins.
Thank you. What are your thoughts on multi-omics data integration and specifically the role of pQTLs in biomarker discovery and target identification?
Yeah, absolutely. And this is, as you can imagine, very rapidly evolving, Stacy, in that the answer to any biological process likely does not lie in any single measurement, whether it be a protein or a metabolite or a lipid or even a genetic sequence, but rather being able to take multitudes of information and put that all together is absolutely critical, which is why we not only perform metabolomics and lipidomics, but also proteomics and RNA sequencing services here at Sapient. In that way, we are able to interrogate multiple areas of dynamic biomarkers and then ultimately statistically integrate these on the back end. Now, you mentioned pQTLs, and what these refer to are proteins for which we have a genetic variant within the human genome that associates with the level of that protein in blood. Now, these can be really quite insightful in providing quite a bit of knowledge regarding not only how genetic variants that have been implicated in disease processes may be acting, but also help us begin to understand how certain proteins may be effectors of biological processes. We can use such statistical tools as Mendelian randomization, which allow us to assign or infer causality even around these circulating proteins using interactions between genetics and protein levels in these pQTLs. And again, this can help identify new biological processes that underlie disease, as well as identify new potential targets for therapeutic intervention.
Thank you. We have a few more that we can go through.
Sure.
How do you view the role of discovery proteomics versus RNA sequencing and target identification?
Yeah, absolutely. Now, if we look across the entirety of the proteome within tissues, now not so much in blood, so there's less correlation in the blood between protein levels and RNA sequencing. But if we look within tissues, for many proteins, there's a reasonable correlation. When the RNA level is high, the protein level is high. Now, there's specific subsets of proteins, whether they be signaling molecules or membrane-bound molecules, where that correlation is well less established. And this has to do with the turnover and the handling and how these proteins are regulated within the cell. In those cases, we find that there's a relatively poor correlation between the degree or the level of the RNA molecule and its subsequent protein level. And the regulation occurs not from the RNA level, but from an alternative mechanism. Now, what's critical here is being able to measure not only the RNA species, but also the protein levels. And for instance, for membrane-bound proteins that are targeted by ADCs, this is a classic example by which RNA sequencing provides limited information regarding the actual copy number of the protein on the membrane, which is essential for developing these drugs. And once again, why we view having multidimensional data or multi-omics data for both proteomics as well as RNA sequencing can be quite elucidating.
Thank you. You mentioned Olink. In regards to Olink, have you done a comparison relative to Sapient's mass spectrometry methods? And can you please walk through the difference in shared measures?
Yeah, absolutely. So Olink is one of the affinity-based approaches, again, that uses two antibodies, whether they be polyclonal or monoclonal, to bind a particular target and then uses a DNA tag that overlaps in the PEA assay to read out the levels of those proteins. Now, when we look at the Olink 3000 Explorer methodology, for which the majority of the data is available from the UK Biobank, and takes the same samples and run Sapient proteomics for our core method that's measuring about 4,000 proteins, we find that there's about 1,000 or such proteins that are overlapping between our systems, where Olink is measuring them and we're measuring them, and we can do comparisons around the quantitation of those specific 1,000 proteins. Which means there's about 2,000 to 3,000 proteins that are unique to each of the systems, whether it be mass spectrometry or whether it be Olink. And this is why one of the main reasons we view this as a complementary interrogation. So even if you have a sample that's been analyzed by Olink, measuring it with mass spectrometry will add on several thousand new proteins. Now, when we think about those proteins that are enriched in each of these systems, again, because we're capturing nanoparticles, or we're using nanoparticles to capture exosomal and membrane-bound proteins, we find that there's many more membrane-bound proteins within our core methodology than are sort of apparent on an Olink or affinity-based approach. But what Olink measures well is very, very low abundant proteins for which amplification is required for adequate signal, including some inflammatory cytokines. And this is why, again, these may be complementary approaches whereby mass spectrometry can provide some of the answers, and then supplementing it with an affinity-based approach can provide answers regarding other proteins that are of great interest.
Thank you. I think we have time for one more.
Sure.
Can you please describe some applications of proteomics specifically within the context of oncology and cancer therapeutics?
Yeah, this is sort of an open-ended question in that there's many, many, many different applications. I'll just review a couple of them, including highlighting some of the ones that we just showed in the application notes. So for instance, in target identification, and this is a key component of the oncology development cycle, what are the right targets to go after, and how do I target a tumor cell specifically without affecting a non-tumor cell? And this is an area within the ADC world or with T-cell engagers that have become quite important. And using proteomics allows us to understand tumor-associated antigens that can be targeted, again, for selectively introducing chemotherapies or immune reactions in order to target the tumor cell without affecting the normal cell. In the instance of PROTACs and degraders, as we showed, proteomics can help us develop these assets and being able to measure the comprehensive nature of the proteome and understand what's being selectively degraded and ensure that other proteins or pathways that are of importance to the cell are not degraded. And again, to limit the adverse reactions that may occur from any of these drugs. We can also use proteomics to screen hundreds to thousands of degrader agents, and in doing so can help in the optimization of the chemistry of these systems. On the back end, as these tools are being developed, proteomics provides a great tool in order to understand target engagement, PK/PD around these various agents, adverse events, companion diagnostics, and ultimately be able to predict who's going to respond to a various therapeutic agent.
Great. That is the last of our questions. Thank you so much, Mo, and thank you to everyone at Sapient, as always, for a wonderful presentation. We appreciate it. And to all of our attendees, we look forward to seeing you at an AAPS eChalk Talk again soon. Thank you
Thank you so
again.
much. Thank you, Stacey. And if anyone would like to contact us, our contact information is provided here on our slide. You can feel free to email us at discover@sapient.bio. There's a Calendly link, as well as our website is located here at the bottom, www.sapient.bio. Thank you again to the AAPS and to Stacey for inviting us for this talk today.
Thank you again, Mo. Everyone have a wonderful day.